This video is all about DNA replication. So let's get started with part 1 of our gene expression video series. So when we're talking about DNA replication, that's going to be the process of making more copies of DNA. But before we can get into all those nitty-gritty details, of which there's a lot, we first need to do a quick refresher on DNA and really talk about what are those features of DNA and how is it the same or different in prokaryotes versus eukaryotes. So remember that all of the DNA that contributes to the well-being of the cell, we call that collectively the chromosome. And so in prokaryotes, which are much smaller and kind of more simple than eukaryotes, okay, so in a prokaryote, they contain one single chromosome that is circular. The DNA is double-stranded, okay, and chromosomes contain essential genes. That means each of those genes are needed for life. Those genes are going to code for proteins that allow the cell to function properly and therefore survive. Okay, so with bacteria, which again is where most of our prokaryotic conversation kind of goes, it's all about a single circular chromosome made up of double-stranded DNA. Okay, now some bacteria have little bonus features, and we did talk about this in our cells conversation. But remember, kind of an accessory thing that some bacteria have are plasmids, and these are extra chromosomal. That means they are outside of the chromosome. So what does that mean? Well, I just told you that the chromosome has all of the essential genes needed for life. So if the plasmid is separate from the chromosome, then that means it does not contain essential genes, okay? So all the genes found on the plasmid are bonus features, okay? They are not necessary for life, but they typically offer the bacteria some sort of advantage. It may offer, antibiotic resistance against penicillin. It may code for a toxin, which is going to help make that pathogen, a little bit more dangerous. It'll weaken the host a little bit more. Maybe it provides the bacteria with a capsule, right, which we talked about previously. So again, these plasmids are bonus features. So if you take a look at this top picture here, you see this particular species, Bacillus cereus, it just has that chromosome. Okay, again, double-stranded DNA. It's overall circular structure, and there's a single chromosome. So there it is. That's all Bacillus cereus has, is the chromosome. But if you look over here at Bacillus anthracis, not only does it have that chromosome, which has all those essential genes, but it actually has 2 plasmids. And notice one of those plasmids codes for a toxin, and then the second one codes for a different toxin. Okay, and so Bacillus anthracis has a chromosome and 2 plasmids. Now, if the bacteria were to lose those 2 plasmids somehow, or it just didn't have them, then it would still be able to live and do its thing. It wouldn't be quite as dangerous to us because it wouldn't have those toxins, but it would still be able to survive because, again, plasmids contain non-essential genes. Now, when we think of eukaryotes, our chromosomes are linear, and we have, depending on the eukaryote, like humans, we have multiple chromosomes, okay? Because eukaryotic cells, remember, are a bit larger, a bit more complex. Some eukaryotes, like humans, are multicellular, right? So it's just a lot more complexity there. But the general features of DNA that we're going to talk about on the next slide are the same, but for eukaryotes, the chromosome is linear, okay? But it's still, that DNA is still double-stranded and supercoiled. So let's kind of dive into more of those details, shall we? We shall. All right, so remember, DNA, deoxyribonucleic acid, is, as I mentioned, our genetic material. And DNA and RNA are both nucleic acids. That's actually what the NA stands for in their abbreviations. So nucleic acids in general are made up of nucleotides. And you can see the structure of the nucleotide in the top right. Nucleotides all have a phosphate group, a sugar, and a nitrogenous base. Okay, so RNA is also made up of nucleotides, but they're a little bit different than the nucleotides that make up DNA. So we're going to focus our conversation right now on DNA. So notice that the phosphate group and the sugar here, okay, these are going to be identical for all the nucleotides that make up DNA. Where the variety comes in is this nitrogenous base, because there are 4 possible nitrogenous bases: adenine, thymine, guanine, and cytosine. And we normally abbreviate those A, T, G, and C. So when we talk about this sequence of DNA, we always just refer to the nitrogenous bases because that's what differs. So we might say, if we look at this picture over here, this one strand of nucleotides, notice the sequence here is A, C, T, and G. Okay, so again, we refer to only the nitrogenous base because that's what's different. For all of these nucleotides that you see in this image here, the phosphate groups are identical. The sugar is identical. What changes is the base. Okay, so that's normally what we refer to. So if you can take a look at either of these pictures right here, you're going to notice that DNA is double-stranded. That means it's made up of 2 strands of nucleotides. Okay, so I was just going through this first strand that you see on the left that had the sequence A, C, T, and G. But notice over here, there's a second strand, and its sequence goes T, G, A, C. So it is double-stranded, okay? DNA, you may have heard, is a double-stranded helix, and that's because it is wound like a helix. So the sugar and the phosphate group make up this backbone, okay, that you can see where my mouse is moving. This backbone is known as the sugar phosphate backbone, and the bases project inwards, okay? So a lot of times we compare the structure of DNA to a spiral staircase. The railing that you hold on to would be that sugar phosphate backbone. The steps would be the nitrogenous bases. Now, when we think about these 2 strands of nucleotides, okay, there are 2 important features of this. Each strand has directionality. Okay, they run in opposite directions. So we say these 2 strands are anti-parallel. So think about like downtown Austin. You're going to come to a one-way road where you're driving from left to right. Well, at the next intersection, right, it's going the opposite direction. Cars are moving from right to left. That's the same gist as this anti-parallel strands. One moves left to right, the other right to left. But we refer to the ends of a DNA molecule as either the 5' end or the 3' end. Okay, so if you look at this left-hand picture, notice on the left strand, it runs 5' to 3'. Okay, but the other strand goes the opposite direction. Okay, so the 5' is now at the bottom on the right-hand side, and it runs up to the 3' at the top. So a lot of times we'll just say one strand runs 5' to 3', the other runs 3' to 5'. Again, they're just opposite of one another, okay? We also say that these 2 strands are complementary. And what that means is these bases in the middle here will do what's called base pairing, okay? So adenine will always pair with thymine. So look at this top example. Do you see how adenine on the left is pairing with thymine on the right? Well, no, now go down 2 nucleotides and notice the thymine on the left is pairing with adenine on the right. That's always going to be the case, okay? Adenine and thymine, think of them as besties. They're always going to be together. Likewise, guanine and cytosine are always going to pair together as well, okay? So Whenever you have an adenine on one of your strands, you're going to have thymine on the other. If you have cytosine on one strand of nucleotides, you're going to have guanine on the other strand. Okay? So again, when we're thinking about DNA, think about the two strands that make up that DNA molecule. The two strands are anti-parallel, meaning they run in opposite directions, and they are complementary because of base pairing. Now, I want to take 1 minute to just give you some extra detail because you might be going, why on earth do we call one end the 5' and one end the 3'? So I'm not going to test you on this, but I just wanted you to know why we call it that. So check out this left-hand image and look at the very first nucleotide. So do you see how there's the phosphate, the sugar, and then the base adenine? Well, because this is the, in this picture, we're looking at it top to bottom. So because this is the first nucleotide, the topmost nucleotide, what do you see at the tippity top of this strand? A phosphate group, right? And this phosphate is attached to the 5' carbon, okay? So when you're looking at the sugar, do you see how at each little point here, remember that if you think back to your chemistry days, at each little point, that's a carbon molecule. So we always number our carbon molecules. So we have 1, 2, 3, 4, and here's our 5th carbon. That's where the phosphate is attached. So we call it the 5' end because that phosphate is attached to our 5' carbon. If you come all the way down to the bottom of this left-hand strand, what is the most bottom thing, excuse me, that you see? Well, here's our little, our sugar here, and our OH group is coming off the 3' end, okay? And so same thing, if you look at the opposite strand at the very bottom where it's 5', you see a phosphate group again, located on the 5' sugar. And then at the top here, on the 3' sugar, you see the OH, okay? So that's just why we call it 5' and 3'. So just one more reminder, okay? DNA is a really long molecule. Thousands of bases make up a DNA molecule, thousands and thousands, okay? So little segments of DNA, a string of those nucleotides, are going to make up a single gene, and that gene is going to code for a specific protein. So there are lots and lots of genes on one piece of DNA, okay? All right, so let's go ahead and apply what we know and think about, well, actually, I think, let me see, am I getting ahead of myself? Please hold. Yes, okay, sorry. All right, so why do we care about the two features of DNA, the fact that the strands are anti-parallel and complementary? Well, by knowing the details of one of those strands of nucleotides, we can deduce the information of the second strand because they're anti-parallel and complementary. So let me show you what I mean. If we know that this top strand runs 5' to 3', then we know the second strand must be opposite of that. So we're going to have our 3' end on the left and our 5' end on the right. Okay? Now, what about our sequence, the DNA sequence? Well, let's say we know that the 5' to 3' strand has this sequence, and I'm not going to read all of those to you. Well, we now can figure out the sequence of that bottom strand of nucleotides because of base pairing. Remember, adenine always pairs with thymine, so we're going to expect to see thymine right here. Next, we have guanine in the 5' strand, so that's going to pair with cytosine. So we'd expect to see cytosine second. And lo and behold, that's what it is. Okay, so notice if whatever's on the 5' to 3' strand on the top, we can use our base pairing rules to figure out what the sequence is on the 3' to 5' strand. And so this is essentially what's going to happen during DNA replication. Okay, by knowing the directionality and sequence of one strand of DNA, the cell can build a second strand of DNA to create an intact, complete molecule. So now we're going to go ahead and apply what you know. So if I tell you that one strand of DNA is 5' T, C, A, T, G, C, G, G, A, T, and then the 3' end, what is the directionality and sequence of the second strand that comprises that DNA molecule? Think back to what we just did. Go ahead and give it a shot. Try it on your own. I suggest you try it from your brain first, then go back to the previous slide and, make any adjustments as needed, and then press play and hear me walk through it with you. All right, so here we go. So we know that this strand runs 5' to 3'. Okay, so we need to start writing the directionality of our next strand. So let's start with the left-hand end. The strand we're given is 5', so that means the missing strand must be 3', right? Because that's opposite. Now let's tackle the other end of the strand of nucleotides. With the given strand, it has a 3' end, so that means our missing strand should be 5', because again, they're just opposite. Now we need to fill in the middle. So this is where the two strands being complementary come into play, all right? So I'm going to go ahead and show you that sequence, but let's talk through it now. So our first nucleotide was a thymine. Thymine always pairs with adenine, so that's going to be what's on the bottom strand. Our second nucleotide in our top strand was cytosine. That always pairs with guanine. Okay, adenine always pairs with thymine. I'm going to go on down a little bit further. Guanine always pairs with cytosine. All right, y'all, that is going to be it for this video. Please let me know if you have any questions.